Right. I hope everyone can hear me. I think I'm loud enough. Yeah, so I'm going to talk about the oligonucleotide sequence confirmation in quality control. And as I'm heading the analytical research and development department, it's a bit of a journey from research and development in our group to the regulated quality control environment. And first of all, I want to introduce the company I work for. So we are a CDMO, founded 27 years ago in Frankfurt. By now, we are having 630 employees approximately. It changes every day, I think. So I was number 180, so we more than doubled in the last 5 years. Yeah, we're doing everything from R&D to commercial oligonucleotide products, and we're certified by the FDA and the Japanese authorities for GMP. Yeah, starting with oligonucleotides, I want to show on this slide how diverse oligonucleotides are. So there are so many modifications on oligonucleotides, and that's just a bit of the modifications we are working with. So you can modify the sugar in the backbone for stability reasons or for the recognition for different enzymes. The bases, you can conjugate your oligonucleotide for pharmacokinetic and drug delivery reasons. And most of the people working with oligonucleotides might know the backbone modification with the phosphorothioate linker instead of a phosphate linker. And as diverse as the modifications are the applications of oligonucleotides in research and therapy. You all might know the mRNA vaccine from the last few years, but there's many more like aptamers binding to receptors or proteins. Then we have antisense oligonucleotides, silencing oligonucleotides, and end of last year, the first single guide RNA-based product was marketed and accepted by the FDA. It's called Casgevy from Vertex Pharmaceuticals. And this is also the reason I'm going in the very end to talk about guide RNA sequence confirmation using mass spectrometry. For everyone who does not know how a single guide RNA looks like, so it's the fusion of the tracer and the cRNA used in CRISPR-Cas9 systems. We have, I hope you can see it, a DNA binding site here, the 20 oligonucleotides in the very beginning on the 5' end, and then we have a Cas9 binding region, which are the 80 remaining oligonucleotides in the end. I also show it here. So this makes the sequence and the structure that comes from the sequence very important and very crucial for the efficacy of the therapy. And that's why we need to control it in the quality control department. So what we normally do, or what you have to do, is the identity by the molecular weight or the molecular mass, and you have to check the molecular sequence, which can be done by next generation sequencing or Sanger sequencing, but it's also possible to do it with mass spectrometry. Other critical quality attributes are purity or the biological activity of the single guide RNA when in the RNP complex with Cas9. So to give a quick overview about the oligonucleotide mass spectrometry and quality control, I want to show where we get samples from. And this is something we need to consider in our research and development department, or at least in our method development, because the sample prep is the first step we need to look at. So it can be that we have high salt concentrations out of the production department, so we need to desalt it, or we have high concentration, we cannot just measure, we need to dilute it, or it's from any matrix. So we have to perform some extracting work. The samples we are looking at are in-process controls, release, and stability samples, depending on, in our case, on what the customer wants, but also what's the best for a good separation. We have to choose our chromatographic system. So the most common is the ion pair reversed phase system, but you can also use size exclusion chromatography or ion exchange chromatography coupled to an normally UV and UV detector and a mass spectrometry detector. In some cases, we also use fluorescence analysis. When running the chromatographic separation, we try to determine the purity, but with oligonucleotides, it's hard to say, okay, this is the purity of the oligonucleotide because there are so many oligonucleotides co-eluting, especially for the longer oligonucleotides. It's hard to separate, let's say, oxidations or so-called N-1, so a truncation of only one base. It's not always possible to do that with chromatography, so we need mass spectrometry to say, okay, this is our main peak, what lies beneath that. And then normally we can assign the peaks quite quickly, but in some cases we need to go further and use tandem mass spectrometry for the sequence analysis, and we also have sometimes isobaric sequences, which is then not the product we want, but has the same mass, and you cannot see it in the MS1 dimension. And that brings me to the sequence confirmation or an introduction to oligonucleotide tandem mass spectrometry. And as for peptides, we have some points in the molecule where the oligonucleotide can break when applying an energy to the oligonucleotide. So these are parts of the oligonucleotide, and just for everyone who does not know how it looks like, if the charge remains at the 5' end, you call these ions A, B, C, or D ions, and if the charge remains on the other side, on the 3' end, it's W, Z, Y, and X ions. This nomenclature is according to Scott McLuckie. He invented this nomenclature in the early 2000s, and this is how we annotate our spectra. And to get a nice fragment ion spectra, you have two ways. Either you can do it with the classic tandem mass spectrometry approach, so you select the precursor, fragment the oligonucleotide, and assign all fragment ions to it, or if the sample is pretty pure, you can also use in-source fragmentation. And just comparing those two techniques, which we are both using at BioSpring. So in-source dissociation, so you have the whole sample, you have not selected anything, so you get a whole MS spectrum, while with tandem mass spectrometry you can select one precursor and then apply an energy to it. Increasing the energy, some ions might fragment and give fragment ions, which you can then use for sequence analysis. And at some point, you have the optimal conditions found for sequence confirmation. How it looks like, so we did some work on the BioAccord where we used the cone voltage ramp for in-source dissociation, and we have an Bion IMS Q12 and Xevo G3 in the QC lab. Both or all instruments are GMP qualified. So what you see here is what I showed on the last slide. So increasing the voltage, gets you to an optimal fragment ion spectrum, which I show here in the middle. But if you go beyond that, you might have overfragmentation and you might lose some information of the sequence. And we assessed that together with Waters in an application note, with the Manchester team. So if you're interested in it, you can clearly go to the website and read it in more detail. And yeah, something Christopher and I did last year is to see how can we speed up our development work. So, and there are two key takeaways we have found out. So the lower the charge state, the higher the CRT voltage needed. I think it's the same for peptides, but we just used the Waters standard. It's a chromatographic standard from 15 to 35 nucleotides. And as you can see on the left-hand side or in picture A, the lower the charge state, really the higher the CRT voltage is that you need to fragment the ions to get the whole sequence covered. And the longer the oligonucleotides get, the higher the CRT voltage is needed at a given charge. And it's not that easy because if the charge is too high, intrinsic fragmentation can also occur, which is one of the problems we have with single guide RNAs, because they are pretty big with 100 nucleotides, they are highly charged and they can fragment right away, although you don't apply any collision voltage to it. So how to get to a sequence cover or a sequence confirmation of single guide RNA, and there are a few ways to get there. One presented, I think, last year by Chris Crittenden from Genentech. He had the latest Orbitrap technology or something like this. And what he did, they did top-down analysis, but they only got to about 80% of the sequence covered. And what we need for good quality control method is at least 80%, so we cannot use it. There's another paper from Alexandre Goyon, also from Genentech, and they do the bottom-up approach. It's like for proteins, you digest your oligonucleotide using enzymes, separated using LC-MS, and then you confirm the sequence of the digestion products. There's one bit older approach. It's a degradation-based analysis, but this one is really, really hard to apply to single guide RNAs. What we do in our lab is the bottom-up approaches, and we have a variety of enzymes which we can use. So Alexandre Goyon described the RNase T1A and also the U2 in their paper in Analytical Chemistry 2021. Eric Wolf from New England Biolabs, he investigated the human RNase 4 initially for mRNA analysis, but we also applied it to single guide RNA because we saw that this enzyme might have beneficial attributes for our oligonucleotides. And then there are more, let's say, approaches which are more for characterization work. One is the approach from Chin et al. From last year. So they blocked the part of the sequence and then used RNase T1 to digest the oligonucleotide. So you have larger chunks of your oligonucleotide, which then can be characterized much easier than if you have really small digestion products. And there's a paper from Yan et al. Where they use RNase H and direct-in oligonucleotides, and we also applied this to the single guide RNA. So in IPRP analysis, you can say the longer the higher the retention time, the longer the oligonucleotide. Approximately, no, it's not 100% that way. But with the RNase H, you can choose where the enzyme cuts the sgRNA. And we had an approach where we cut the single guide RNA into 4 pieces and had 100% sequence coverage, which is really good, and that for, I don't know, 20 or 30 different oligonucleotides, so we had a quite good method. With the RNase T1, we have small oligonucleotides, and sometimes we lose some sequence information. So if you don't know where your digestion product comes from, where do you want to assign it, it's impossible. So you might lose some information using the RNase T1. And the human RNase 4 is somewhere in between, and is now used as the new standard in our lab, as long as we are not using the RNase H as it is in research and development stage at the moment, but we want to bring it to GMP analysis this year. And to also show you some mass spec data, because we are at ASMS, so this might be interesting for you as well. So the RNase T1, as I said, produces small oligonucleotides, and if you fragment it and you see 35 volts is pretty high energy to fragment an oligonucleotide. But one advantage of these fragment ion spectrum is per base, you have a lot of fragment ions you can assign to. So the, if you calculate your fragment ions, you can almost find all of them. Using the RNase H approach, where you have longer oligonucleotides, you might not have every single fragment ion for every single base. So you have a lower fragment ion to base ratio here. But still, with both approaches, you can at least assign 100% of the sequence from the digestion products, but maybe not for the whole single guide RNA sequence. So this is all coming from R&D, and now what my supervisor always asks is, how can we get it to GMP analysis? Because that's what the customers ask for. And first step is automatizing the sample preparation. So we have the Andrew Alliance in the lab doing recurrent sample preparation steps. Then for feasibility studies, we are using intact mass, just confirming the digestion products, looking for cyclophosphates and phosphates in the digestion products. And last but not least, we're using confirmed sequence to look at the sequence covered after tandem mass spectrometry experiments. And with that, I just want to sum up a little bit. So for oligonucleotide sequencing, you can either use the BioAccord or the QTOF systems. We have both in the lab, and we are happy with both options, depending on the oligonucleotide length, of course. What we always do is adjust the fragmentation energy, so that we're not having overfragmentation and that we can use it for GMP analysis. For single guide RNAs, the bottom-up approach is by now our favorite approach. And the enzymes, of course, strongly determine the output sequence coverage. And you have to look on the sequence of your oligonucleotide. So if it's G-rich, I would avoid the RNase T1, which cuts after on the 3' end after G. For direct sequencing, we're not there yet, but we may be there in the near future. We were also checking on the possibility to use IMS or cyclic IMS for top-down sequencing, and showed in a few slides. We have a poster with Walters on that topic on Wednesday. But in the end, I have to say there's still some research ongoing, and we have to do some more research. And I don't think it's boring for me and my team the next few years. So good for me, and hopefully also good for the customers we work for, that we are doing some more research. Speaking about the team, without them, couldn't be standing here talking about the oligonucleotide sequence confirmation. Christopher will give a talk on Tuesday on bioanalytics, and the team is quite big, and they always do what I can to give me the opportunity to talk here. And also a big thank you to the Waters team in Germany and in England, because they helped us a lot with setting up everything. Right, then just something for the ASMS this year. We have 2 posters, so Christoph and I, we're standing right next to each other, so you can't miss it. If you find one of us, you find the other one. Christoph will talk about bioanalytics, and I will talk about the investigation of parameters in IP-RP separation. And on Wednesday, there's a poster from Catherine Tremblay on the cyclic eye mobility for top-down sequencing of synthetic guide RNAs. And with that, I know I'm the last thing between you and the reception. Don't want to make it longer than it takes. Thank you for your attention.